So then we start with the last meeting, and I think first of all we're going to start with Nina Raben, who will give a presentation on skeletal muscle abnormalities and Pompe disease can be reversed by enzyme replacement therapy. And I'm very happy to hear that, Nina, so I'm very anxious to hear your talk.
Thank you. So it looks like we now all agree that the currently available therapy reverses cardiac abnormalities but has limited effect on skeletal muscle. And in very general terms, the ERT in late onset patients slows disease progression. In infantile, one may think that the therapy would convert this most severe phenotype into milder late onset phenotype, but in fact it created a new clinical phenotype. So what's the problem? Why skeletal muscle is so resistant to therapy? First, it's muscle mass, and there is nothing we can do about it. There is immune response to enzyme replacement therapy, and you've heard about immunomodulation. There is also inefficient muscle targeting because of the properties of the enzyme the same, not quite optimal for lysosomal targeting. And that is exactly the reason why there is a next generation of ERT. There is also, and you probably saw it on many slides, that there is a degree of pre-existing pathology. And I think that it is a little bit of a gray area here, unless there is an extreme case, like for example here, when muscle is almost completely destroyed, and in this case it is really perhaps not reversible. So, but in general, we are looking at the basic biology of skeletal muscle damage, and I will talk about it in the first part of my talk, and in the second, our experience with Amicus recombinant enzyme. So we all know there is no question that the primary defect, lysosomal storage, glycogen accumulation, expansion of lysosome, lysosomal rupture, or also, in addition, lysosomal membrane permeabilization, plus neurological abnormalities. But what leads to relentless muscle damage in Pompe disease is a cascade of the pathogenic events. And today I will focus on 3 of those. First, autophagic accumulation, and I'll be won't spend too much time on that. A, because I presented it many times for this audience, and B, because it's finally very well accepted as the secondary abnormality in Pompe skeletal muscle. I'll talk about the 2 major kinases responsible for signaling pathway. And the reason we are interested in it is because these 2 kinases define, in many sense, the well-being of the cells, the balance between the synthesis and degradation. They're involved in control of protein synthesis, and both of these kinases are a major regulator of autophagy. And finally, I will touch upon our new data not published on the metabolic defect in Pompe skeletal muscle. And this, I have to say, is new data. And to me, they're mostly interesting because the traditional dogma for many years, we always thought that the lysosomal glycogen accumulation and the lack of the release of glucose from the lysosome back to the cytosol does not have any metabolic consequences for the cell. At some point, it was even taboo to touch this subject. So anyway, there will be some, I will present some new data on the metabolic abnormalities. So autophagy, very briefly, major intracellular degradative pathway starts with the formation of a double membrane, engulfs portion of the cytoplasm, leads to formation of autophagosome. Autophagosome fuse with lysosome, where the content of autophagosome is digested, and the products, the building blocks, are released to the cytosol. fundamentally, it is a response to starvation. So when nutrients are not available, the cell digests its own components. That's why it's called autophagy, self-eating, to sustain the cell to survive until the situation becomes better. It is also shown that the system works even when at the time of plenty, when the nutrients are available. And in this case, the system serves as a housekeeping function. It allows the cell to get rid of old or worn out organelles, including, for example, mitochondria. What happens in Pompe disease, there are two things which we found in Pompe disease. More autophagosomes are made. We have a clue why this happens, but this part is not completely clear why there is an induction of autophagy. But we do know, and from our experimental data, that there is a problem between fusion of autophagosomes and lysosomes. And when these two structures cannot fuse, the vesicles become to build up, and this results in major autophagic abnormality, accumulation, piling up of all kinds of vesicles with undigested cytosolic content. So the consequences of this autophagic buildup, which you can see here by electron microscopy of skeletal muscle in knockout mice, is accumulation of potentially toxic proteins, which are normally should be eliminated through this pathway, but now they sit in the muscle, accumulation of aberrant mitochondria and oxidative stress. So this is shown by electron microscopy. The autophagic buildup can also be detected in isolated single muscle fibers stained for autophagosomal and lysosomal markers. And as you can see in the core of muscle fibers, that there's a huge accumulation of lysosomes and autophagosome. This is in knockout Pompe mice, and it's essentially exactly the same in human. These are muscle biopsy from Pompe patients, and you can see autophagic buildup by electron microscopy, by immunostaining, and actually, when you know what to look for, you can isolate single muscle fibers, fix them, or live muscle fibers, and look under the regular bench microscope, and those bubbles which span along the whole length of the fiber represent the area of autophagic buildup. Okay, now let's move to signaling. MTOR kinase, mammalian target of rapamycin complex 1, is a major negative regulator of autophagy. So this mTOR kinase is also master regulator of protein synthesis, and it is directly involved in the control of muscle mass. Therefore, it's very relevant to what's happening in Pompe disease, because muscle atrophy is a hallmark of the disease. These are in old mice. You can appreciate the difference in size of muscle fibers between the knockout at the bottom and the wild-type mouse. So when mTOR is active, it phosphorylates its downstream targets and stimulates protein synthesis. In addition to that, mTOR phosphorylates and inhibits autophagy initiation, kinase, and inhibit autophagy. So this system makes perfect sense. When the cell grows and synthesizes protein, it doesn't want to eat itself. So autophagy is inhibited, protein synthesis is proceeded. What is a recent development in the lysosomal field, that this activation of the mTOR kinase happens at the lysosomal membrane. When the nutrients are available, mTOR moves, is recruited. The process is so much more complicated. I will try to simplify it as much as I can. So mTOR moves from the cytosol to the lysosomes, and it needs to do it in order to be activated by a protein called Rab, which resides on the lysosomal membrane. Now, during starvation, when the nutrients are not available, another complex comes to the lysosome. It's AMPK kinase and THC complex. MTOR moves away from the lysosome. The downstream targets are not phosphorylated. Synthesis is inhibited. So AMPK, while at the lysosome, inhibit mTOR activator. In other words, mTOR activates autophagy indirectly by inhibiting mTOR. On the other hand, AMPK also activates autophagy directly by activating the same autophagy initiation kinase, but phosphorylating it at different sites. So what happens in Pompe skeletal muscle? We found that there is excessive accumulation of AMPK and THC complex at the lysosome, and that there is a diminished mTOR activity. Number 3, metabolome profiling. So here we're looking at the metabolome, which are products of the basic biochemical pathway, which are extremely conserved during evolution. So what we found in our Pompe mice are very likely to be the same in humans. Here is the principal component analysis showing the difference between the two groups, knockout mice and the wild type. And you can see there is a significant difference between these two groups. And this one is a heat map of hierarchical cluster analysis, again, comparing these two groups, showing individual samples. We had here 6 knockout mice and 6 wild type. And as you can see by the difference in the colors, that these two groups are quite separate. So the second part of my talk, we tested the Amicus recombinant enzyme, which is now called AT-GAA, and we directly compared it with the myozyme. We already heard that the difference between the enzyme, the increased number of mannose 6-phosphate group on the enzyme, and therefore the enzyme has much better targeting properties. So in our first series of experiments, and we published it, what we showed was that this new enzyme, Amicus enzyme, is in every measured outcome in Pompe mice was much better than the current myozyme enzyme replacement therapy. We looked at GAA activity, at glycogen clearance, muscle strength, muscle fiber size, and also lysosomal expansion and autophagy. And frankly, I was mostly impressed with the effect on autophagy, because in all my long experience with preclinical studies with myozyme, autophagic buildup remains on ERT with myozyme, no matter what I've done, no matter how long I treated these mice. So, in fact, to my very pleasant surprise, 40% of fibers after therapy with Amicus enzyme looked completely normal or near normal. As you can see, this is the wild type, and this is the ERT knockout treated muscle fibers, and there is no autophagic buildup, and the green dots is using the lysosomal marker LAMP. So the remaining fibers, 60%, still contain autophagic buildup, but they contain autophagic buildup of different sizes, as if it was a snapshot, and you can see slowly disappearance or reduction in size of the autophagic buildup. Encouraged by this data, we conducted a long-term studies starting with mice of 3 to 4 months of age. We did 11 injections, so overall course was for 5 months, and they were 8 to 9 months old at the end of this study. So we did the grip strengths, and it seemed that it's perfectly fine, even though I have to say I'm not a big believer in this test. Glycogen level was all the way back to the wild type level, perfectly normal. So what we then did, we looked at the lysosomal and autophagy markers, at the MTOR and AMPK signaling, and on the metabolic profile of muscle. So we wanted to see whether this new enzyme can reverse not only autophagy, but also the downstream events, the signaling and the metabolome analysis. So what is it? We are looking at markers. So we're looking at markers, autophagical lysosomal marker, LAMP. Autophagic marker, we look at the marker which are responsible for different stages of autophagic process. Like 3 of them, I'm not going to burden you with the names, but some of them at the stage when the double membrane is only formed, the next stage when the autophagosome is formed. We look at the classical autophagic marker, LC3, which is a protein which stays on the lysosomes all the way until it fuses with autophagosome. And we're looking at the P62, which is by itself an autophagic carga and a very good indication of the autophagic process. I'm not even asking you to look at all those graphs. I will tell you that the bottom line is that all of them, except for one, AT-GAA7, at this stage, and a little bit of LC3, all back to the wild-type level. LC3, which is a classical marker for autophagosome, also goes down, but not completely to the wild-type level. And indeed, this time, after 5 months on therapy, we see 70 to 80% of normal or near-normal fibers, as you can see in the knockout, lysosome, enlarged lysosome, autophagic buildup, and here normal, perfectly normal. I'd say very close to normal fibers. And again, you can see it by a regular microscope of live muscle fibers. Most of them are like this, and some contain a very short areas of autophagic accumulation. So now we also used one novel marker, which may be very useful for other investigators, and this is a marker called galactin-3, which is a marker of lysosomal damage. And what we consistently see that there is a significant increase in skeletal muscle of this marker in knockout, and it's completely reversed back to wild-type level in after Amicus treatment. We've done additional studies with galactin, and it looks like that the increase is really linked to the presence of autophagic buildup. Unfortunately, we could not detect it in blood because that would be really a good biomarker, but we do see it in the skeletal muscle. Now, with the signaling pathway, again, it's a complicated pathways, but the bottom line here that we are looking at the levels and phosphorylation level, meaning the activity of AMPK, autophagy initiation kinase, downstream target, upstream regulator of this kinase. And the bottom line is that all of them, with the exception of one, TSC, all back to the wild-type level. We now look at MTOR, and again, we look at MTOR itself, autophagy initiation kinase, its phosphorylation, downstream targets, and upstream regulators so that we can see the whole pathway. And again, most of it, as I mentioned, diminished mTOR activity, which we found in the knockout mice, is partially, not completely, but partially reversed on the enzyme replacement therapy. What about the metabolome? This PCA analysis, you can see after enzyme replacement therapy, this group are right in between the knockout and the wild type, suggesting that there is a significant improvement there. And the same with the heat map. Now the knockout on ERT look like intermediate between these two groups. Now, we look at the individual pathway in the metabolome, and what we see is the increase in glucose, in glycogen precursor. UDP glucose is a precursor of glycogen. This suggests that the glycogen synthesis is inhibited. After ERT, we see an improvement on this. We look at the glycolytic pathway. I'm not asking you to look at all of this, but what we do see that glycolysis is down. There is a decrease in glucose 1-phosphate, glucose 6-phosphate, and several glycolytic metabolites. They are all improved on enzyme replacement therapy. We're now looking at the Krebs cycle, and what we see is that glycolysis is downregulated in the knockout, but TCA cycle metabolites are increased despite the reduction in glycolysis. And we also see an increase in acetyl-CoA. This suggests, to put it in a nutshell, that muscle relies on fatty acids for energy because sugar is limiting, which means that the accumulation of glycogen and the absence of acid alpha-glucosidase, which degrades lysosomal glycogen, and the glucose is released back into the cytosol, has serious metabolic consequences for the cells. In support of this, there is an increase in carnitine, which transport long-chain fatty acids to mitochondria. On ERT with Amicus Enzyme, the metabolites are largely back to the wild type, although carnitine remains elevated. Energy. There is an energy
Nina,
deficit.
could you end your presentation almost?
That's it, half a minute, yeah. There is an energy deficit, which is back to wild type levels on the therapy. And we do see an increase in total amino acid as well as individual amino acids, indicating that there is an increased proteolysis in Pompe skeletal muscle, which is a very well-known phenomenon. And we don't see major changes in ERT-treated mice. So this is the way we see pathogenesis, lysosomal enlargement, autophagy, mitochondrial abnormalities, abnormal signaling and metabolic defect, glucose starvation, and that the new enzyme significantly improves essentially all aspects of the pathogenesis. One thing which I would like to point out, we started therapy at 3 months of age when there is the autophagic buildup is full blown, but the mice are completely asymptomatic at this stage. We also looked at the 6-week-old mice, and we already see autophagic buildup. We also tried to see with the autophagic and lysosomal marker at 10-day-old mice, and you already see the beginning of autophagic buildup. So the bottom line is, if we can start with a good enzyme in asymptomatic, when pathology is already there, we can have almost complete cure of the disease. Thank you very much.